At midnight, a male copepod crosses black water, where another body may be hundreds of lengths away. The ocean around him appears vacant, yet a faint chemical trail bends through the moving darkness. He turns into that invisible path, following evidence left by a female he cannot see. This search captures a central pelagic mystery: how scattered animals meet before currents erase every clue. In open water, distance is not measured only in meters, but in fading signals and limited energy. A missed trail can mean hours of fruitless swimming, greater exposure to predators, and no reproduction. Some animals release scents. Others flash, vibrate, call, gather, transform, or wait beside predictable landmarks. Each solution answers the same problem under different constraints of depth, current, vision, and population density. The meeting must also occur at the right season, developmental stage, and reproductive condition. Natural selection favors signals that travel far enough without becoming impossibly expensive or dangerously conspicuous. Yet, no single strategy makes the ocean crowded, and every signal can fail before reaching a receiver. Researchers, therefore, study courtship as a sequence of detection, approach, recognition, contact, and successful spawning. At every stage, turbulence or deception may separate two animals that briefly occupied the same moving world. The mystery is not that animals possess miraculous senses, but that ordinary physics sometimes carries sufficient information. Our investigation follows those fragile messages from their release to the rare encounter they can produce. Seawater transports dissolved molecules, pressure disturbances, sound, and light, according to different physical rules. Chemical cues can persist as filaments rather than forming a smooth, widening cloud. Small crustaceans. Sampling such filaments may compare encounters across time and adjust their swimming direction. Laboratory observations show male copepods tracking female-associated chemical trails with rapid turns and looping searches. The behavior links a cue to action. Trail contact prompts pursuit, while loss prompts local casting. This adaptation narrows a three-dimensional search, and the consequence is a greater chance of overtaking the female. Turbulence can stretch the trail, break it into patches, or fold it across the female's actual route. Tracking, therefore, works within a limited time and distance, not as an infallible underwater map. Many marine worms and other invertebrates solve distance differently by synchronizing spawning in dense seasonal gatherings. Day length, temperature, tides, lunar illumination, and biological clocks can help narrow the reproductive window. When many individuals release gametes together, dilution remains severe, but timing raises local concentrations dramatically. The verified principle is convergence in time, even when adults remain dispersed for much of the year. Reef fishes often converge in both time and place, visiting promontories, channels, or current-swept spawning sites. Visual displays and rapid ascents then coordinate partners at ranges where light still carries useful detail. If you watched from meters away, the gathering could seem sudden, despite weeks of physiological preparation. Below sunlight, bioluminescence provides another channel, created through chemical reactions within organisms or microbial symbionts. Species-specific flash patterns are documented in several ostracods, whose males produce luminous courtship displays. A male releases timed points of blue light, creating a moving pattern that females can approach. The cue prompts orientation. The repeated pattern supports recognition, and darkness gives the display strong contrast. Its consequence is efficient signaling, though conspicuous light may also attract predators or unintended competitors. Sound travels especially far underwater, allowing some large animals to advertise beyond visual or chemical range. Baleen whale songs and calls participate in reproductive behavior, although their precise information content varies among species. Long-range sound reduces search space, but hearing a caller does not demonstrate that mating followed. That distinction separates a plausible communication function from direct observation of reproductive success. Tagging and passive acoustic recording reveal movements and calling, while genetic parentage can test eventual reproduction. These methods illuminate separate links, yet rarely capture the entire sequence in remote open water. A recorded call shows production. A responding movement supports attraction. Offspring genetics establishes successful parentage. Without those connections, researchers avoid treating every seasonal sound as a mating invitation. Some deep-sea fishes use an extreme solution in which a tiny male permanently attaches to a female. In certain ceratioid anglerfishes, attachment can develop into tissue fusion and shared circulation. The cue action sequence begins earlier when the male detects chemical or sensory information and searches for a female. Contact prompts biting. Specialized anatomy sustains attachment, and the consequence is reproductive readiness when eggs mature. This strategy evolved in particular lineages and should not be generalized to all deep-sea fishes. Other anglerfishes mate without permanent fusion, reminding us that similar darkness does not dictate one outcome. The rarity of direct encounters also selects for bodies that can store sperm after a successful meeting. Sperm storage separates mating time from fertilization time, buffering uncertain access to partners. Hermaphroditism can likewise increase the reproductive value of encounters in some marine groups. It does not guarantee self-fertilization, and many hermaphroditic animals still require another individual. Sequential sex change can align reproductive roles with body size, social position, or local group structure. On reefs, documented social cues can trigger sex change when a dominant individual disappears. The cue is altered social structure. The action is physiological transformation, and the consequence restores breeding roles. This solution depends on recurring groups and differs from the challenges of truly solitary pelagic animals. At hydrothermal vents, habitat patches act like islands where suitable chemistry concentrates animals and potential mates. Finding the habitat can indirectly solve the partner problem because both sexes respond to the same environmental beacon. Currents carrying vent chemicals and larval settlement cues may guide dispersers toward these rare productive patches. Evidence is strongest when settlement patterns, experiments, and sensory responses agree. But long-distance navigation remains difficult to observe. Floating objects, seamounts, ice edges, and fronts can also aggregate food and animals without serving as deliberate rendezvous signals. Researchers must distinguish active mate, searching from simple concentration around shared resources. An apparent pair may reflect feeding, migration, or common current transport, rather than courtship. Behavioral sequences and reproductive condition provide stronger evidence than proximity alone. Scale changes everything, because a millimeter-long animal experiences viscosity, turbulence, and signal decay differently from a whale. For plankton, one body length can contain detailed chemical structure that larger swimmers cross almost instantly. For whales, low-frequency sound can bridge broad distances, but travel time and shifting collars complicate localization. You can imagine each species inhabiting a different sensory ocean layered within the same seawater. Predation further shapes those layers because bright, loud, or persistent advertisement exposes the signaler. Some animals reduce risk by signaling briefly, displaying at twilight, or courting within shelter. Others produce many signals because only a small fraction will reach an appropriate receiver. Energy imposes another filter, especially where food is scarce, and prolonged search reduces future survival. A successful strategy must gain enough encounters to offset its metabolic cost and mortality risk. This trade-off explains why passive waiting, seasonal aggregation, and permanent attachment can each be locally effective. Climate change can alter the physical channels that once made encounters predictable. Warming shifts distributions and breeding times, potentially separating partners that formerly overlapped. Changing currents can redirect chemical trails, larvae, and migrations, while noise can mask biologically important sounds. Ocean acidification may affect sensory processes in some species, but responses vary and broad claims remain premature. Artificial light can disrupt natural darkness near coasts, changing the visibility of courtship and predators. Fishing can remove large adults or skew sex ratios, reducing mate availability beyond simple population decline. When density falls, reproduction may decline disproportionately because remaining individuals rarely locate compatible partners. This process, one form of an allele effect, can slow recovery even after direct mortality decreases. Protecting spawning grounds and migration corridors therefore preserves meetings, not merely bodies. To understand those meetings, future studies must connect detected signals with movement, contact, fertilization, and offspring. Miniature tags may reveal fine-scale approaches, but attaching instruments can alter the smallest animals. Passive acoustic arrays can triangulate callers, although identity and reproductive outcome still require independent evidence. Chemical plumes remain transient, and a detected molecule rarely reveals which individual responded. Genetic parentage across seasons can measure success, but it reconstructs outcomes rather than witnessing courtship. The strongest conclusions will combine a few complementary methods while acknowledging missing links. If you return to the copepod's trail, its apparent simplicity now contains turbulence, timing, and sensory decisions. Researchers can test how often males intercept trails, how long filaments persist, and which turns improve pursuit. Field observations are needed because laboratory tanks simplify currents and artificially compress distances. Comparable work across species can reveal whether scarcity favors stronger signals, better tracking, aggregation, or flexible reproductive anatomy. It may also show when population loss pushes an encounter system past a critical threshold. No observation will make every hidden meeting visible, and unexplained movements should remain unexplained. The next decisive evidence will follow a signal continuously from one known animal to another. It must then connect that encounter with fertilization or parentage rather than assuming success. Until then, many ocean courtships remain supported by partial sequences, strong mechanisms, and honest gaps. The male copepod still turns through darkness, not toward certainty, but toward a fading trail that sometimes makes reproduction possible.